Good morning, students. We are discussing about the principle and processes of biotechnology. In the last class, we have discussed about the rDNA technology. Now, see the rDNA technology involves following steps. First is the isolation of genetic material, that is DNA, fragmentation of DNA by restriction endonuclease, isolation of desired DNA fragments, ligation of the DNA fragments into a vector, transferring rDNA into host, culturing host cells in a medium at large scale, then extraction of desired product. Isolation of genetic material DNA. Most of the eukaryotic cells have DNA as genetic material cells. To form rDNA, the DNA should be pure, that is, free from all biomolecules. DNA located inside nucleus enclosed by plasma membrane. Genes intertwined with histone protein. Cell have a break open to release DNA and other macromolecules like RNA, proteins, polysaccharides, lipids, etc. Cells are lysed by lytic enzymes, for example, lysozyme for bacteria, cellulase for plant cell, and chitinase for fungus. Macromolecules treated with specific enzymes to remove RNA, ribonuclease, proteins, protease. Purified DNA precipitate with addition of ethanol appear as collection of fine threads in suspension. See the DNA extraction. Cells are lysed using a detergent that disperses the plasma membrane. Cell content are treated with protease to destroy protein and RNase to destroy RNA. Cell debris is pillated in a centrifuge. The supernatant containing the DNA is transferred to a clean tube. The DNA is precipitated with ethanol. It forms viscous strands that can be spooled on a glass rod. See the picture how DNA is spooled out. Cutting of DNA at a specific location. Purified DNA, that is source DNA, desirable gene DNA and vector DNA, incubated with specific restriction enzymes for the process, that is digestion at optimal condition. Progression of restriction enzyme digestion is checked. Agarose gel electrophoresis. DNA negatively charged, so move towards positive electrodes. Once gene of interest and cut vector with space for gene of interest is isolated, DNA ligase is added. DNA ligase acts as a glue. This step is preparation of recombinant DNA. See the all process through a picture. Digestion with the restriction enzymes, the plasmid and the foreign DNA, then electrophoresis, then ligation of gene of interest and the vector DNA, that is plasmid. Amplification of gene of interest using PCR, that is polymerase chain reaction. PCR technology used to amplify a single or a few copies of piece of DNA to generate thousands to millions of copies of particular DNA sequence in vitro. In vitro means in lab condition. Components required: primers, small chemically synthesized oligonucleotides that are complementary to the region of DNA, nucleotides and DNA polymerase incubated together. Steps in PCR: denaturation of double stranded DNA by heating. A single stranded DNA formed. Primer is added, binds to complementary region, that is annealing. DNA polymerization with the nucleotides in medium. Genomic DNA acts as a template. Replication repeated many times 1 billion copies of desirable DNA. Amplification involves thermostable DNA polymerases from the Thermus equiticans bacteria to facilitate replication even at high temperature. See the steps of polymerase chain reaction. Region to be amplified, that is shown in the red color. The double stranded DNA is denatured, changes into single stranded in presence of heat, then addition of primers, that is annealing, and then DNA polymerase, in presence of DNA polymerase and deoxynucleotides, that polymerization reaction starts and forms the double stranded copies, that is extension, and that cycle is repeated many times and forms the amplified 1 billion times and forms billions of copies of the DNA. Insertion of recombinant DNA into host cell or organism. Insertion of rDNA possible by making host competent, achieved by chemical treatment, microinjection, ballistic and gene gun method or disarming pathogens. Phenotype of organism alters when rDNA is transferred to host. For example, rDNA with gene resistance against antibiotic ampicillin transferred to E. coli host. Phenotypes alters resistance against ampicillin transformants. When plated on agar plate with ampicillin, transformed will grow and non-transformed die. Presence of ampicillin resistance gene select transformed cells and considered as selectable marker. Obtaining the foreign gene product. Alien DNA binds with the cloning vector, rDNA transferred to the suitable host, bacteria, plant or animal cell. rDNA expresses itself target protein under appropriate optimal condition and after cloning or of rDNA to a particular number of copies. Target protein produced in large scale. In heterologous host, if protein encoding gene is expressed, recombinant protein. Heterologous host genes or gene fragments that does not naturally present in host, the gene expresses itself, recombinant protein. Cells which harbor cloned genes of interest grown in laboratory in a small scale. Culture of dividing host used to extract desired protein, then purified by different separation technique. In large scale, cells can be cultured in continuous culture system. Here the used medium is drained out from one side and fresh medium added from other to maintain the cell physiologically most active and exponential phase. The culture method produces a large biomass leading to higher yield of desired protein. Biomass, biological material derived from living or recently living organism. To produce large quantities of products, bioreactors are used and the capacity is about 100 to 1000 liters. Large volume of culture is processes. Bioreactors, vessels in which raw materials, recombinant organism like microbial plant, animal and human cell and culture medium with ambient condition, biologically converted into specific products like protein or enzymes. Optimal growth is achieved with growth conditions, temperature, pH, substrates, salts, vitamins and oxygen. The structure of bioreactor. Commonly used stirring type, cylindrical with curved base to facilitate mixing or content, equipped with stirrer, even mixing and oxygen availability throughout reactor, oxygen delivery system, bubble air through reactor, agitator, thorough mixing of contents, also have foam control system, temperature control system, pH control system and sampling port. Sampling port withdraws small volume of culture periodically to check the progression. See the picture of that bioreactor, simple stirrer tank reactor and aspersed stirred tank bioreactor through which sterile air bubbles are aspersed. Downstream processing. Downstream processing refers to the recovery and purification of biosynthetic products, particularly pharmaceuticals, from natural source such as animal or plant tissue or fermentation broth. Products obtained from bioreactors subjected to series of processes before marketing as finished product. Processes include separation and purification of biological products. Products are formulated with suitable preservative and then should undergo through clinical trials like drugs. Strict quality control testing is required for the final biological products. Process of recombinant DNA technology.